everybody, welcome to Ms. McGuire online class, and we are covering the cellular level of organization. Okay, let's go ahead and begin. So we're going to talk about cells in this video lecture, and we're going to start with the structure of cellular membrane. And you see over here that a membrane, cellular membrane, is mostly phospholipids, right? It's a phospholipid bilayer. That means that phospholipids are organized in two rows. And phospholipid is a molecule with very interesting structure. The structure is shown over here on the bottom of the slide. So the phospholipid, it has a head and a tail, and the head is hydrophilic. It has charge, so you can see oxygen over here has a charge, so it attracts water, so it's a water-loving part. And a tail is hydrophobic, and hydrophobic means they repel water. These molecules do not dissolve in water because they are neutral, they don't have any charge. And because this unique structure, when you put phospholipids in a fluid, in a watery substance, they tend to form a double layer. When the tails are facing towards each other and heads are facing water, right? That's why the membrane is made of this this bilayer because of the chemical properties of phospholipid, right? So you can see that even from this small diagram, you can see that the major function of cell membrane is separation of internal environment of the cell from external environment. Right, so it's provides the boundary for a cell. It defines a cell. It keeps cells, organelles, and cytosol inside and separated from extracellular fluids. So when we look at cell membrane structure in a little bit more detail, we see these phospholipids, right, over here, and how they form this double layer or bilayer, and how this form boundary and a barrier between cytoplasm and extracellular fluid. And yes, phospholipids are major lipids. They about 75% of all the lipids that make up cell membrane, right? But we do have other lipids involved. 20% is cholesterol, and cholesterol on this diagram shown with this, yellow molecule over here. It's 4 rings, right? A very distinct structure of the cholesterol, and cholesterol increases membrane stability and fluidity. And 5% are glycolipids. Those are lipids with polar sugar groups on the outer membrane surface. So you can see these short carbohydrate chains, oligosaccharides, what we call, right, when they are not very long. So these oligosaccharides attached to phospholipids, some of them, forming these glycolipids. And actually all the sugar covering on the surface of cell membrane is called a glycocalyx. Right, so are we doing good here with cell membrane, right? So cell membrane is phospholipid bilayer. Phospholipids are about 75% of all the lipids, but we do have cholesterol and glycolipids as well. Right, and then we have proteins, all right? So proteins are shown here in this bluish color, And proteins can be divided into integral and peripheral. So integral proteins firmly inserted into the membrane, and most of them are transmembrane. That means they kind of go through the whole this bilayer, right? So it's that's why we call it transmembrane. That means they go through the whole membrane. And those proteins are usually channels, because they form this channel and allow molecules, ions to move through them, right? So those are integral and transmembrane proteins. Peripheral proteins are loosely attached to integral proteins. So if you look over here, we can see over here that is peripheral protein that's pretty much kind of attached only to the internal surface of the membrane. Right, this one, it's not shown on this diagram, but we can have peripheral proteins that attach to this transmembrane protein. So let's look at the previous parts, like this one, for example. Right, this protein is attached to transmembrane protein. So that's a peripheral protein, or over here on the top of this one. And I just mentioned glycocalyx. Glycocalyx consists of sugar, carbohydrates sticking out of a cell surface. So if this sugar are attached to lipids, we call it glycolipid. If it's attached to protein, it's a glycoprotein. And glycocalyx is really important in a cell-to-cell recognition, in the communication between your immune system and your own cells. So your immune system can recognize this pattern of glycocalyx and understand, as well as it, it distinguishes your own cell from some foreign cells. So glycocalyx has extreme importance in a normal functioning of the cell. Right, so that's a membrane proteins. Okay, so here, this diagram shows us a function of membrane proteins. So they can be, as we already mentioned, protein channels. So let's see if they show here any protein channel. Well, they don't, but they show you just here, right there, right? So this is the protein channel, and function is transport. So because we have, sometimes large molecules or even small molecules, but or ions, because they have a charge, they cannot move through your membrane directly. So they need a protein for their transport. And this transmembrane protein, this one, for example, is just open or leakage channels. So pretty much ions can move down their concentration gradient constantly. So they can leak in and leak out of the cell, right? This one shows us active transport. So this protein will only transport molecules or ions inside or outside of the cell with the use of ATP. So that's an active transport. We call those proteins pumps. But nevertheless, the function of both of these proteins is transport, right? They also can be receptor proteins. So for example, if you have like some hormone that is signaling a molecule, we also call it ligand. This molecule can attach to its receptor and it will produce some signal transduction inside the cell, and maybe, tell your cell to, maybe divide or start synthesizing something or breaking down some large molecules, right? So that's signaling, signal transduction, and function is receptor protein. Also, proteins can be enzymes, right? So it shows over here that, you know, reactants attached to active site of the enzyme, and those proteins catalyze, excuse me, reaction, either synthesizing or breaking down molecules, right? So proteins can perform enzymatic function. They also can create junctions between your cells, like shown over here, so intracellular junction. We already mentioned that it can be glycoproteins, and then the function will be cell-to-cell recognition. So either, your neighboring cells can recognize, maybe, like white blood cells can recognize some cells, right? Or maybe cells of your immune system can recognize your own cell. So cell-to-cell recognition. And they also can function in attachment to cytoskeleton and extracellular matrix. So you can see over here that these transmembrane proteins are attached to the cytoskeleton inside and extracellular matrix outside, and keeping your cell in a fixed position, right? So let's review again. What are the function? Attachment, receptor, transport, intracellular junction, cell-to-cell recognition, or enzymatic activity. Okay, so because we have this membrane, right? Every single cell has a membrane. So if you wonder, what are the organelles that every single cell has, it would be cell membrane, DNA, cytoplasm, and ribosomes. So it doesn't matter what cell is that. We even have like bacterial cells that are prokaryotic cells, but any cell that is a living cell will have cell membrane, DNA, cytoplasm, and ribosomes. So because every single cell has a membrane, and the membrane forms a barrier between internal and external environment, cell, and we just mentioned that we have this membrane protein that allow transport, right? So now we're pretty much gonna look what kind of transport can be achieved through a cell membrane, right? So the transport can be divided into passive and active transport. And passive transport doesn't require ATP, and active transport does. So if ATP is not used through the movement of ions or molecules across the membrane, we can call it passive. Now, on this diagram, you see this is outside, this is inside, and plasma membrane. So passive transport can be simple diffusion through the lipid barrier by a lipid bilayer, when small non-charged molecules pretty much moving just through this phospholipid barrier, simple diffusion. And they move down their concentration gradient from high concentration to low concentration. No ATP is used. We also can have facilitated diffusion. And facilitated diffusion means, well, something gonna facilitate this process, right? And what gonna facilitate is a protein, right? This transmembrane protein that we call protein channel. And this one, for example, is non-specific. So that means pretty much many different ions and molecules can move through, but most of these proteins are actually specific. So this is facilitated diffusion through a specific transporter. That means if this is glucose transporter, and only glucose will go through. But it's still passive transport because molecules or ions moving from high concentration to low concentration without ATP input. And another kind of type of passive transport is osmosis. And osmosis is not much different. The only difference is that only, water can move by osmosis. So it's a movement of water from high concentration of water to low concentration of water. And water can pass through a lipid bilayer directly, right? But most of the water molecules pass through the special proteins that we call aquaporins, right? So that's passive transport. Now, if you look at active transport, then we have what we call pumps. We use ATP energy, your cells use ATP energy. So what do we need? We need a protein, right? And we need ATP, and those pumps gonna transfer now substances, in these examples, sodium and potassium ions against their concentration gradient, right? From low concentration to high concentration. So that's why you kind of need this energy. You need to push something that you have already high concentration of it, so you need to push even more, right? So example of active transport here is sodium-potassium pump, shown right there. So it's pumping sodium out, potassium in, and exchange is 3 sodium out and 2 potassium in. And it's used ATP directly. So that's called primary active transport. We do have secondary active transport that's shown right here next to our primary. And the difference is, if you look at this protein that kind of like pinkish color, I would say, you see there is no direct use of ATP. However, this transport directly depends on primary transport. So first, this primary transport, this sodium-potassium pump, gonna pump sodium out. It's gonna create high concentration of sodium in extracellular fluid, and then sodium obviously wanna move down its concentration gradient, but doing it, it takes glucose with it. So glucose is moved inside the cell by co-transport, right? So it's a secondary transport and it's co-transport. So glucose would not move on its own, but it does because it follows the sodium. And in this example, sodium moved down its concentration gradient from high to low, and glucose will move against the concentration gradient. Right. So now, if we have some drugs that would, stop this pump from functioning. So if you disable this sodium-potassium pump, then the secondary transport shuts off as well. When the over here, when you see this glucose by passive transport, you don't need any sodium-potassium pump for this facilitated diffusion of glucose to happen, right? But transport glucose by secondary transport require ATP pump. I hope it makes sense, right? So even we don't have direct use of ATP, this movement still depend on a amount and presence of ATP inside a cell. Right? Another example of active transport include endocytosis and exocytosis. And endo and exocytosis are movement of particles inside endocytosis or outside exocytosis of cells inside the vesicles. And vesicles are formed from this plasma membrane. So here we have 3 types of endocytosis: phagocytosis, pinocytosis, receptor-mediated endocytosis, and exocytosis on its own, right? So let's look at it. So here we have, let's go and start with our pinocytosis. So in pinocytosis, vesicles form at the plasma membrane and bring fluids and small molecules into cell. Very often, this process is called cell drinking, Right, so you can see over here, it's just a small little vesicle is formed from the membrane and stuff are moving inside the cell within this pinosome. So example would be waters and small molecules, as we just said, right? Substances involved extracellular fluid with dissolved molecules such as nutrients. That's a pinocytosis, so movement inside a cell within the vesicle formed from cell membrane, but it will be fluids and dissolved substances, right? And this does require ATP. Now go phagocytosis. Now we have a bigger, sometimes solid particles, right, moving into the cell. And they call it cell eating, in parentheses, of course. Example would be how bacteria or viruses or cellular debris or other foreign materials are taken in by white blood cells. Right, so phagocytes. So here's the cell, it's formed what is called pseudopods, like a little extension of the cytoplasm, and it encloses the bacteria forming the phagosome, and this bacterium is taken inside, right? So large particles are brought into the cell by cytoplasmic extensions called pseudopodia that engulf the particles and pull it into the cell, right? So also active transport require ATP. And another example of active transport is receptor-mediated endocytosis. So the substances are moving inside a cell, also in a form of vesicles that form from cell membrane. The difference over here, we need a stimulus. We need to initiate this process with some signaling molecule, right? So the target molecules bind to the receptors, and those are receptors on the protein. So it's a receptor protein on a membrane substance. And this is triggering a vesicle formation. These molecules that are binding to receptor proteins are called ligands. And example would be cholesterol, ions, that are moved inside of cells through receptor-mediated endocytosis. So that's our active transport. Okay, so, and this one is exocytosis, right? And exocytosis is movement of the particles outside the cell, and it's easy, can be continuous secretion. So you, this diagram from the Golgi, vesicles are formed from Golgi apparatus, and whatever the cell is secreting, right, this vesicle moves towards the cell membrane, it fuses with the cell membrane and release it, those substances out of a cell. Or it can be regulated secretion. When vesicles are formed, they move towards the cell membrane only when we have some signaling molecule, some ligand attached to a receptor protein. So we have ligand attached to receptor protein and it trigger this regulated secretion. For example, this is how calcium can trigger some secretion of endocrine glands. Okay, so that was exocytosis. I always need to move myself, always on the way. Okay, so that was kind of very brief introduction to a cell membrane. And now let's talk about what is inside, what is surrounding by a cell membrane. So on this diagram, you can see typical animal cell, and here we have a membrane, cell membrane, and inside this pretty large organelle shown here is a nucleus. Now, everything between nucleus and a cell membrane is called cytoplasm. So cytoplasm, you see here, equals cytosol, that is a liquid portion of cytoplasm, and all the organelles between cell membrane or plasma membrane, that's the same, and nucleus. So if you look here, you see mitochondria, you see endoplasmic reticulum, ER, rough ER, you see smooth ER, you can see Golgi apparatus. Of course, I already mentioned the nucleus. Then it's maybe some vacuole, peroxisome, lysosomes, centrosomes, microtubules that are part of the cytoskeleton, free-floating ribosomes over here. Right, so all these organelles, they have their specific structure and they perform their specific function within a cell. So here we have like very brief overview of major function of organelles. They might have some additional functions, but just for simplicity here, let's just mention at least one or two functions of organelles listed on this slide. So vacuole, one of the functions is storage. Ribosomes are site of protein synthesis. So ribosomes are actually synthesized proteins, and this function is so critical and so important that every single cell pretty much that exists on this planet has ribosomes. Not every single cell has nucleus, but they all have ribosomes. That tells us that protein synthesis is essential function of cells. Then we have ER or endoplasmic reticulum. We have rough ER and smooth ER. And the difference is they both have membranous structure, and in the rough ER, ribosomes are attached to it, making it look, giving it this rough appearance. And smooth ER do not have ribosomes. And because rough ER has ribosomes, right? So it's ER with ribosomes bound to it. And because ribosomes synthesize proteins, then the function of rough ER is to assist protein synthesis. And specifically, those proteins that will be part of the membrane or the ones that will be secreted out of a cell. So secreted proteins or membrane proteins. Smooth ER involved in a lipid metabolism. So synthesis of the lipids and catabolism of the lipids. That's a function of smooth ER. Smooth ER does not participate into protein synthesis because it's lacking ribosomes. Golgi apparatus sorts and packages proteins that it receives from rough ER, right? So ribosomes synthesize proteins. They usually deposit it inside the lumen of rough ER, inside those cisterns, and then vesicle is formed from a rough ER, and those vesicles move to Golgi apparatus first. And then from Golgi apparatus, or within Golgi apparatus, proteins are sorted, they modified, they stored, and secretory vesicles formed from Golgi now moving towards the cell membrane, and either those proteins became part of cell membrane or they secreted out of the cell. So mitochondria, the major function of mitochondria is ATP production. I don't know, it might be some other function because might not be. I never heard about any other function of mitochondria, but who knows? right? But function over here is production of ATP. Now, lysosomes digest foreign substances and worn out organelles. Centrioles help during cell division. They help in formation of a mitotic spindle during meiosis. So they pretty much kind of help your cell to divide. Nucleus, control center, it holds your DNA. Nucleolus is a part of the nucleus that is the site of ribosome synthesis. And cell membrane, we already talked about function, and it's the outer membrane of a cell, and it controls the movement of substances in and out of cell, and it creates boundaries. It's defined as cell. There is no cell membrane, no cell, right? Okay, so here, just the kind of like quickly, we're looking at another organelle like cytoskeleton, considered to be also organelle within a cell, and it provides structural support made of microtubules, and those microtubules can be kind of bigger and smaller in a diameter, right? So over here, we can see different proteins that are part of cytoskeleton, like actin, tubulin, right, keratin. So that's part of the cytoskeleton. And another organelles, such as cilia and flagella, allow for movement. Either the whole cell is moving, for example, the sperm cell is moving because of the flagellum, and cilia, in human body, we do not have cells that move by cilia, but we do have cilia that move substances across a cell membrane. For example, your respiratory tract is covered by epithelium that is ciliated, pseudostratified columnar ciliated epithelium, and this cilia moves a mucus across a cell membrane from your respiratory tract towards your oral cavity, and then you can swallow this mucus with trapped bacteria or some maybe some debris from the air, some dust, whatever get into your respiratory tract. So cilia and flagella are for movement or mostly transport of materials. Also, proteins that are part of the cytoskeleton allow movement or contraction of the cell. Like, for example, your skeletal muscles contract because you have actin and myosin filaments, those protein inside your muscle cells, and when they contract, the whole muscle contract, allow your muscle to move your bones, and that produce movement of skeletal muscles. Okay, and organization of the nucleus and DNA. So your DNA is located inside the nucleus because you are eukaryotic organism. The nucleus itself has a double membrane that called nuclear envelope, and there are pores that allow substances to move in and out, such as, like, for example, messenger RNA. And DNA is a pretty huge molecule that need to be packed inside the nucleus. And that's why there is kind of like several levels of DNA packaging. So if you look on this diagram, you can see that that's your DNA, that is double helix, right? So this is nucleic acid, right, made of nucleotides. So all these letters A, C, G, and T, they represent building blocks of the DNA. They called nucleotides. All right, so this is our kind of like structure of DNA. And then DNA, instead of being loose and this huge and loose DNA, it's packed very tightly around special proteins called histones, forming nucleosomes. And then this nucleosome condenses more and more and more, making, your chromosome. So chromatin is pretty much still pretty loose DNA, but the difference between chromatin and your DNA is that chromatin also includes those histones, right? So when DNA packed around histones, but it's still kind of loose, we call it chromatin, but when it condenses and assumes this, very familiar structure, then we call it chromosome. So chromosome is condensed chromatin. Chromatin is DNA and histones and a little bit RNA, but let's just keep it simple, right? So again, what is chromosome? Chromosome is condensed chromatin. What is chromatin? DNA and histones packed in these nucleosomes, right? And now when we have this nice packaging, then, we can fit everything inside nucleus. Another thing is you can see chromosomes and they become visible only when cell is ready to divide. The rest of the time, when cell is not dividing, we cannot see chromosomes, but we see a loose chromatin. Okay. Now, this slide kind of tells us about how your cells divide and how they differentiate into all types of the cells that make up your body, like neurons, skill cells, red blood cells, heart muscle cell. So we all start with one single cell, right, that we call zygote. This has happened when ovum is fertilized by sperm. So we have the first zygote. And this first zygote, when it starts dividing, so the few divisions, those cells have ability to differentiate into every single cell in your body. So those cells are called totipotent cells. So at the very, very beginning, when zygote just goes through just few divisions, those cells are totipotent. In a human embryonic stem cells, they are pluripotent. They can still differentiate into all the cells of your body, right? But totipotent cells, I think they can also differentiate into placental cells, and so they're more potent, right? That's what. So they have ability to differentiate into more different cells still than pluripotent. But pluripotent cells, those are embryonic stem cells. They pretty much can form any cell in your body. Now, multipotent, oligopotent, and unipotent, you see this division tell us that those cells can differentiate into less and less cells, and unipotent can only differentiate in its own kind of cells. So human embryonic cells are pluripotent, and adult bone marrow cells, skin cells, cord blood, deciduous teeth, right? They are multipotent stem cells. And they can differentiate still into good number of different cells. So I think this diagram will explain it maybe a little bit better. So here we're gonna start, we will see how blood cells are formed. So formation of blood cells are called hematopoiesis. So hematopoiesis is differentiation of multipotent stem cells into blood cells. So here you have multipotent hematopoietic stem cells. So this cell not only can differentiate into all these cells that can be found in your blood plasma and in your tissue fluids, it also keeps its own population. So it divides, but it kind of, you don't run out of it, right? They renew themselves. That's what this arrow shows. So they keep their population or number of cells, would say, constant. So, but these multipotent cells can differentiate into myeloid progenitor cells and lymphoid progenitor cells. And lymphoid progenitor cells, so this is multipotent, right? This is not, because myeloid progenitor cells cannot differentiate into, all these cells that are, I would say, come from myeloid, right? So, and myeloid cells that form from myeloid progenitor cells cannot differentiate into lymphoid cells. But multipotent stem cells, they can be either myeloid or lymphoid progenitor cells. And then lymphoid progenitor cells will differentiate into natural killer cells, lymphocytes, and those lymphocytes then become T lymphocyte, B lymphocytes, and B lymphocytes become plasma cells, right? So all these natural killer T cells, B cells, plasma cells can be found in your blood plasma, right? And some of them in your tissue fluids. Now, myeloid progenitor cells give rise to the rest of your blood cells, right? So it gives rise megakaryocytes that form platelets, erythrocytes, mast cells, and myeloblast cells that progenitor for basophils, neutrophils, eosinophils, and monocytes became, let's say, macrophages, dendritic cells. So the point over here that I'm trying to make, and I don't know if you guys get it or not, but we all start with one cell, right? And all the rest of this millions and trillions of cells that make your body, they are ancestor of that very first cell. That means that very first cells, they have ability to differentiate into different cells. So they have ability to become your muscle cell, your nerve cell, your skin cell, your liver cell, and so on, right? And because those stem cells can differentiate into different kind of cells, we call them potent, right? Potency is ability to do something, and they can be either pluripotent. I don't want to mention this totipotent because it's just very, very few cells at the beginning of our life, but they can be pluripotent during embryonic development, or multipotent in adult stem cells, oligopotent, unipotent cells. An example of this cell differentiation and cell formation from the common progenitor cells is hematopoiesis. Right? That's the point. Okay, and by the way, that's a good news right now for some of you. So if you get to this point, I think if you get to this point, slide number 13, then if you in my anatomy class, this is where you can stop. I forgot to tell you that. So the good news happened couple minutes ago. So I'm recording this video for both my anatomy class and anatomy and physiology class. So if you are in anatomy class, that's your last slide, slide number 13. If you already watched, listen me through slide 14 and 15, good. But There is 2 more slides left. This slide 14, 15, 16, 17 for anatomy and physiology class only. That makes sense? Okay. So now this is for you guys if you are in my anatomy and physiology class. And if you're only in anatomy class and you're still watching it, then good for you, because you still need this later when you take physiology class. Ooh, now, what is that? Resting membrane potential. What it's all about. Now look at this diagram. So you see this green structure that represents a cell, and it's represent human cell because we are in human anatomy and physiology class, right? So that's my cell or your cell or any other person's cell. And we are in animal kingdom. So this is animal cell. Animal cells can only stay alive if they're surrounded by fluids. If there is no fluid surrounding animal cell, it will die. An example would be your skin. The outer layer of your skin, stratum corneum, those cells are not surrounded by fluid and they are dead. Right? But the most of your cells that are alive, they're surrounded by this extracellular fluid. Now, so you have fluid inside the cell, right? Intracellular fluid. You have fluid outside of the cell. And it's not water, it's actually solution. So we have ions inside the cell, outside the cells. We have molecules, we have proteins, for example, right? And what actually happened that your cells, they separate charges. So there is separation of charges between the cell and its environment. And this separation of oppositely charged particles or ions across a membrane creates a membrane potential. So it's create a potential energy. So here how to remember it. Your cells are actually little batteries. So if you have a battery that you put in your cell phone or a controller, what is a battery? Battery means you have some, device that separates the charges. You have positive end of the battery and negative end of the battery. So you have the separation of charges. And because you have separation of charges, this battery carries potential energy. So now when you can use this energy, if you connect the ends of the battery and you can use it to power your computer or your remote and so on. So the same happens with your cells. There is a separation of charges between a cell and extracellular fluid. And it also creates a little battery and it creates potential energy. And this potential energy is called resting membrane potential. And we can measure it. So you can measure the amount of voltage a difference in voltage between the intracellular and extracellular fluids. If you measure this voltage, it will give you a range between -50 to -100 millivolts. And cell inside is negative and more negative and more positive outside. So cell is negative inside and positive outside. But let's look at next diagram that will make lots of sense over here. So look, so this is blue line is a cell membrane. This is cytosol, so this is inside the cell. This is extracellular fluid. And membrane is not permeable, it's selectively permeable. So some, substances cannot move through. For example, ions cannot move through the membrane. They need channel proteins. So now if you look inside cytosol, you can see the large number of proteins. And those proteins are negatively charged right over here. So that gives us some negative charge inside. Plus, what else? Look at this, sodium-potassium pump. Sodium-potassium pump found in every single cell of your body. And what sodium-potassium does, it pumps sodium out, you see the sodium out and potassium in. And when it's pumped sodium out and potassium in constantly, sodium out, potassium in, sodium out, potassium in, what do you think gonna happen? Well, what's gonna happen, we will have a lot of sodium outside and a lot of potassium inside, right? Sodium out, potassium in, lots of sodium and lots of potassium. I think potassium is shown here by this orange circle and sodium in the purple. So you see lots of purples outside. So that's a sodium and lots of orange inside. That's a potassium. So sodium potassium pump creates what we call chemical gradient. That means, you know, you have high concentration of sodium outside and high concentration of potassium inside. This is how you remember it. Your cells are salty bananas. So you have banana and you put salt on the top, that's your sodium, and banana is rich in potassium. So that's your banana potassium, and this is your salt. Plus sodium potassium pumps 3 sodium out and 2 potassium in. So you see, you losing more positive charges and gaining less positive charges. So again, it makes it a little bit more negative inside, right? So we have proteins with negative charge, that make it negative. We have less potassium coming in than sodium leaving the cell. So again, if you're losing positive charges, right, then you became more negative. But that's not the rest. So sodium potassium pump creates chemical gradient. Now, when you have a lot of sodium outside and a lot of potassium inside, where do you think the sodium wants to go? The sodium wants to come back and potassium wanna leave the cell, because if you create chemical gradients, if you have lots of sodium here, it's actually gonna move down its concentration gradient. If you have channels, if you have way for those ions to move, and you do, you see, you have this protein, orange protein, and this purple protein, and this is called leakage channel. This is or leak channel. This is leak channel or leakage channel for potassium. So potassium start moving out. Again, potassium moving out, taking positive charges out, less positive inside, more negative inside. And you would say like, hold on a second, but sodium is moving back in. So you're losing positive charges here, but you're gaining positive charges. Shouldn't it like cancel itself out? It does not cancel itself out, because plasma is about 50 to 75 times more permeable to potassium than sodium. So yes, sodium kind of moves in, but 50 to 75 times slower and less sodium moves in than potassium moves out. And these leakage proteins, they create electrical gradient. So sodium potassium pump, chemical gradient, leakage channel, electrical gradient. And what happened, the cells are actually losing a lot of positive charges, became negative inside and positive outside. And when chemical and electrical gradients equals out, right, then we have this resting membrane potential, right? So, and resting membrane potential in this example, it's -70, so it stays constant, because sodium potassium pump constantly pumping sodium out, potassium in, potassium constantly escaping, sodium is moving slowly, right? but all this system works in a harmony to keep every single cell in your body to make, I would say, every cell in your body is this little battery, but you have separation of negative and positive charges. And this allows for many, many cellular activities. And it will be very important when we start talking about your neurons or your muscle cells. So right now, it really introduction to resting membrane potential. So what do we want to remember? The first thing we want to remember is that your cells, they are little batteries, right? Because there is separation of charges across a plasma membranes. Cells are negative inside and positive outside. And because it's a battery, it has potential energy. Now, how do we call this potential energy? We call it membrane resting potential, or just resting potential. And we can measure the voltage of this potential. And if you measure it, it will give you minus 50 to 100, in this example, minus 70. And minus just tells you that cell is more negative inside. And another thing you want to remember probably is that your cell is salty banana, so you have higher concentration of sodium in extracellular fluid and higher concentration of potassium in the cytosol. So salty banana and little battery. And this is possible because of the sodium potassium pump, because of the leakage channel, and the fact that you have way more potassium leakage channels than you have sodium leakage channels, right? I hope it makes sense. This is the last slide. And we will discuss resting membrane potential again. And I know maybe at this point, at this moment, it's a little bit complicated, but it all will make sense. Okay, so that's it for this lecture. Thank you very much for watching, and I hope it was helpful. So I will see you next time.